The efforts and motivation of physicians around the world fighting against this disease continues to provide the Fragile X community with hope for a bright future. It's time to hear from some of these top physicians specializing in Fragile X syndrome to discuss how to quickly and accurately diagnose this illness, as well as potentially effective therapies for this disease.
Okay, well, I'm Ted Brown, and I'm the moderator for this panel today. And really, we have the three superstars of Fragile X research with us. They all each are doing great work, and it's really a pleasure to be able to bring them together today and present some information that should be interesting to everybody. I think that maybe the first person we might call on is Liz, who recently had a really spectacular paper published on improving cognition in Fragile X with a new drug. And this is the first trial that I'm aware of where cognition has really been shown to have a change. So maybe Liz, you could give us a little overview of this study and where it's going.
Sure. Well, so this trial was a trial of a phosphodiesterase inhibitor that raises cyclic AMP in the brain of individuals with Fragile X syndrome, or really anyone. But patients with Fragile X syndrome have been shown to have a deficit in cyclic AMP production, as have the animal models. And this was something that I discovered back in 1989. And so it's been around a long time, but we didn't have drugs that could target this problem because all the drugs had too much off-target side effects in the body. And so the drug that was developed that we used is a brain-specific phosphodiesterase 4D inhibitor. And so it was able to just work in the brain, and we avoided all the side effects. And we did a small phase 2 trial in which we employed some of the outcome measures that we've developed as a result of the previous failed trials, things like the NIH toolbox, which we've worked extensively with David Hessel to validate for intellectual disability. And really, this is the first trial where we were able to use a good repeatable cognitive measure because we had done the validation and developed it. And then we also had measures, parent report measures of the kinds of behaviors that were important to them, as well as some standard measures of behavior and some of the newer biomarkers that we have developed, like eye tracking and event-related potentials. And this trial, often in phase 2 studies, you see a small signal, but we actually had a statistically significant signal on a number of the subtests of the NIH toolbox, as well as the parents rated specific problems that limited their individual Fragile X's function in both language and daily function. And they rated these as better on the drug versus the placebo. And that corresponded to the improvement in the cognitive function on the toolbox. And we also saw some improvement in ERP biomarker. So to my knowledge, this is the first phase 2 study where we've actually seen a performance-based cognitive measure, a parent-rated measure of function, and a biomarker all improve correspondingly in the same study in a placebo-controlled fashion. So we have great hope for this, but we are going to need to be able to show that this drug improves function in a phase 3 trial before we can move forward. And I really think this trial was successful because of all the work in the whole field that we've done in the past that came out of some of our earlier failed trials, where we understood that we needed better outcome measures and better trial designs.
I have many patients chomping at the bit to initiate this study.
Yeah,
so it's great that it could start to occur at multiple centers throughout the country.
And you saw a carryover effect, right? You were going to do it.
So carryover effect with the drug had been seen in the mouse model. And we actually had the study design was a crossover design where patients were randomized to drug or placebo, and then they switched over to the opposite. And so they had 3 months in both treatment periods. And the company was very interested in understanding whether there was this carryover effect in humans. And we did see it. So of course, all of the primary analyses were based on the first period in the study, but we were able to document the carryover effect in the second period of study. So that was interesting because it really corresponded to the animal work.
Great. Craig?
Yes,
Yeah.
and to add, I think that this success may be specific to the mechanism, but I think what we're alluding to is it may really be specific to the development of the field and the improvement in signal detection. And certainly there have been a number of preclinical studies, primarily in the mouse model, some Drosophila, some other models, of phenotypic rescue, animal improved, phenotype that have not translated into humans. But really the tools and the approach that Liz took with performance-based measures, enrolling males, so sort of, winnowing down the population to have a more homogeneous group enrolled. These are important steps where I think we could be successful, which also makes you think that some of our past large trials may have failed to detect signal in certain populations. Things could have been blurred by placebo response. So I think we're really revisiting what we've done in the past with our new methodology as much as we're exploring new potential mechanisms.
We don't want to forget about the girls, because many girls are affected with Fragile X syndrome, and I think they may respond just as well as the males. I think we need to study the girls, but I think that the NIH toolbox, which is a cognitive measure that we didn't have before in other studies, but we will get more data on the NIH toolbox. This is very exciting because it can demonstrate cognitive improvements.
Yeah, and I think we do want to study the girls because I have a lot of girls, of course, and I'm sure Craig does. But I think the key is that we want to study them in separate studies because the girls perform differently, and the amount and type of improvement you may see in the girls turns out to be different from the guys. And when we combined them all together, we just increased our scatter and made it harder for us to see results. And I think that's something we've learned over the years and will lead to, better development programs. So trials in both genders, but separate trials, I think is probably going to wind up being the most successful thing, especially for targeting cognition.
Craig, do you have a comment on that?
Yeah. I mean, the way we conceptualize it now is really thinking about almost a continuum of Fragile X, which of course includes sex as a feature, with females who on average are less impaired than males. And it may well be that different medications or medication combinations based on where you are on the continuum, which can be represented by protein expression, can be represented by what we can measure with cognitive measures, with EEG, with brain activity. And so, when we talk about Fragile X treatment development now, I think of type 2 diabetes and hemoglobin A1C as an example, where depending on how severe your blood glucose is, you may need insulin or 2 oral meds or 1 oral med that's different. So we're really honing down and refining not only how we detect response, but how we detect who exactly with Fragile X may respond, because Ronnie's absolutely right. Women and girls in Fragile X are extremely important, but should they potentially respond, differently to a particular agent, we have to design our studies to detect that and not blur our signal.
And you have a rather novel new EEG assay, right, that looks at phase and the gamma frequency. Can you explain a little bit about that to us?
Yeah, so through our NIH Center grants, we've been looking to really analyze EEG or brain electrical activity in Fragile X knockout mouse study and in human study really in synchrony. And one consistent translational marker that's been noted is increased high frequency or gamma band activity at rest. That's very well conserved across species. And we've now been moving to use this electrophysiologic high frequency marker in treatment studies across mouse and human studies at the same time. And we've shown, we've done recent work with some single drug dosing of a GABA modulator. And we've seen synchronous response in the mouse, in the human, for example, to single drug dosing with gamma band improvement. So we think we can use brain electrophysiology to help bridge this translational gap and maybe help us predict a bit better, again, who with Fragile X may respond to a particular agent. And early on in treatment development, does a specific molecule, a specific drug under study, lend itself well to using EEG in human study, for example, to stratify study inclusion, to have a pharmacodynamic marker of treatment response. There are various ways it can be used, and there's been other advancements with auditory response, and Liz and Lauren Etheridge and others and folks at Davis have been involved. But I think it's more that, can we measure things in a similar manner in animal and human studies? Because look, audiogenic seizures, we can't test that in humans. Marble bearing, we can't test that in humans. There are a lot of behavioral mouse assays that aren't relatively easily translated, but we think electrophysiology is.
Yeah, I think the mouse behaviors don't translate too well to humans. And I think a great example of that is if you're not an anxious mouse, you might be a stupid mouse because you're probably going to get eaten. So, we just don't know what functions behaviors serve in these animal models. But when you get down to the basic cellular science and electrophysiology, that's something that is a basic building block of the brain that may be directly translatable.
The problem is in young children, they oftentimes don't tolerate an EEG cap. And so you have to go, I mean, there's other sorts of developmental testing that you can look at over time. Yeah. And some eye tracking is doable in young kids, and other measures, but the EEG is hard to get in under 5-year-olds.
And we don't want to just restrict this to Fragile X syndrome. We want to discuss also the potential for treatments for FRAXTAS and FAXPOI. So Rondi, maybe you could give us a little overview of where you see that going or where it is today. What's
yes, we've been in FAXTAS, which is the Fragile X associated tremor ataxia syndrome. We definitely need a treatment because this is a neurodegenerative disorder that gets much worse over time in a limited number of older carriers. About 50% of males, about 16 to 20% of females, and the numbers go up higher as they age. So we know there's mitochondrial problems, and there's some new meds out that we think can help to reverse some of the mitochondrial abnormalities. And there's 2 or 3 potential studies that, pending funding, could be tried in individuals with FAXTAS. Perhaps the earlier the better, before individuals lose too many neurons or have irreversible cognitive deficits.
Right. And what about the area of generics or supplements or over-the-counter drugs that might be available? Where do you see that? So we just
published a paper that looked at the fibroblasts in individuals from individuals with FAXTAS, and sulforaphane, which is a dietary supplement that can have some very positive effects, like turning on the internal NRF2 system. It's an internal antioxidant system that hopefully can reverse oxidative stress and mitochondrial problems. So we'll see in an open label trial whether this is helpful for individuals with FAXTAS, and we're just initiating an open label trial now.
And this is a substance that's present in high concentration in broccoli sprouts, as I understand it, right?
Yeah, broccoli and cruciform vegetables, like cauliflower too, and Brussels sprouts. So you got to eat your broccoli and other cruciform vegetables, and then you can get a good dose of sulforaphane.
I might ask the question, what do families want? What do the parents and that dealing with their Fragile X children want mostly, and as they get to be adults? How do you see that? Anyone?
So we actually have done a couple of big surveys of the family population about what are their biggest problems and what would they like. And when you ask them about symptoms, they, in the pediatric population, the families flag anxiety, resolution of anxiety, improvement in the anxiety that's limiting for the patients as their biggest problem. In the older patients, though, cognition really becomes equal to behavior. And I think this is, the patients are growing up and they're no longer in school, and now they can't support themselves. And families really would like the patients to be functional in life and to have enough cognition that they can do something meaningful and have a job. And so that becomes a higher priority for families. The really young families often flag speech because the kids aren't talking yet as something really important to them.
Yeah, cognition and behavior, those are top elements. But, you know, a lot of families are very interested in gene therapy, and we're living in an age where, you know, gene therapy for Fragile X may become available in the next few years. So that's very exciting.
What form would that take, Rondi?
It's either using CRISPR-Cas9 to, you know, fix or replace the gene, or maybe even putting FMRP in a AAV virus, maybe injecting it into the spinal fluid. It's, you know, a lot of different options, but we've seen in, you know, a variety of other disorders where gene therapy is making a difference. So people are getting quite creative about thinking about these interventions for Fragile X.
Exciting. Craig, maybe I could ask you about dealing with the FDA, which is an important area that we all face. How do you see this going forward?
Yeah, I think advocacy with the FDA is as important, if not more important, than advocacy for funding from places in the United States like the National Institutes of Health and the Centers for Disease Control, because we have to build the understanding of the agency and the EMA in Europe and other regulatory agencies about Fragile X. And the National Fragile X Foundation did organize really a stakeholder-driven PFDD meeting this past spring, which I think was along those ends. And, you know, really what we're up against now is as we've improved our ability to quantify performance. Liz has the great example of cognitive performance, other performance-based measures. How can we put forth those kind of measures as primary outcomes when really tradition in the interpretation of Fragile X at the FDA and the psychiatry branch has been based on observer report and measures such as the aberrant behavior checklist and others, that we've had challenges with their use in Fragile X with large placebo response, lack of specificity to the condition. So I think we're at a real key time in our field of expanding our armamentarium of quantitative outcomes. And that is absolutely going to take agreement and partnership with the FDA to move it forward.
I would say absolutely. And I think what's happening is that we have better measures for Fragile X now, but those don't have a long history with the FDA. And many of the standardized cognitive measures were never standardized for people with intellectual disability. They just all fall at the floor, and there's no way to actually measure change with those measures. Yet those are the measures that the FDA has seen in other patient populations that are not as intellectually impaired or are not intellectually impaired at all. And so I think we have work to do all of the validation that is needed to convince the regulators that this is the best measure, as well as trying to help convince them that we have a rare disease and we can't do validation on hundreds of thousands of people. We have to accept validation that's been done on a reasonable sized population, as long as it's well done and shows good scientific correlate with measures that already exist.
And what do you think about personalized medicine in the area of Fragile X? And, you know, are the heterogeneity of patients that we see, or children and adults that have the syndrome, how does that affect where the drug studies should be going?
Well, I think if we appropriately characterize folks that we're enrolling, we then have the potential to do subgroup analyses by sex, by protein expression, by electrophysiology, by performance. And then I think we'll be successful potentially in these subgroups, a third, 40, 50 25% at a time. I think this is the direction we're going. But if we don't determine in early phase studies who may best respond, I think we're really still at risk in phase 3 of blurring the picture in the analysis.
What about drug combinations? Do you think we'll be seeing that more and more, or is that going to be necessary?
That's what happens clinically. As clinicians, we're always using drug combinations, and many people that get enrolled in studies are on different combination of drugs. But I think we have to, characterize the analysis to reflect what are the multiplicity of drugs that people are taking. But we can't expect all patients to respond the same to any new medication. We have to think about, maybe some aspects about background gene effects in addition to the molecular variability that we see that maybe could be quantitated by a very good FMRP measure from blood. I think the subgroup analysis that Craig talked about is really important to think about, particularly with multiple meds on board before they do the study.
Yeah, I mean, I think these things are critical, but also when you think about all of the things that FMRP does in cells, it's got there are a lot of potential mechanisms to target. So one would think, kind of like cancer, usually you're not going to treat a tumor with one medication. You're going to need multiple medications to actually target all the pieces of what FMRP does. And one piece or another of that may be more important in different patients. And so all the characterization possible, as well as options for different patients to respond to different medications, and options to build upon what medication we know works for a certain category of medications and add something else to improve functioning further. I think it's a lot like building cancer chemotherapy, where you get something that works and then you add another thing onto it and you gradually make Fragile X a treatable disease. I mean, from a standpoint of mechanism, because we already do treat the disease. We treat the symptoms of the disease, but making the core problem a treatable disease by adding one thing to another over the years, I think is going to be the way we're going to need to go.
People need to think that FMRP is really a mother controlling protein that controls a lot of different, translation, but also it may be lowered in other forms of autism and other neurodevelopmental disorders. It's found to be low even in schizophrenia. So the targeted treatments that we develop for Fragile X may be helpful for many other disorders too, but it is a really important protein.
Right. And what about the role of CBD? I know there was a recent study that you've all been involved with that's, I guess, going to be continued on another phase 3 trial. What would you say about this role, Rhonda?
We all like CBD. So CBD, a lot of families just get it through the internet, at a hemp website or organic, or, whether there's more CBD than THC in the preparation. And the, most recent control trial of a topical ointment preparation showed benefit in those that had a full mutation that was fully methylated. So there, the division of the patients in terms of characterizing what their methylation status was very important to show benefit. So the most affected that had a full mutation that was fully methylated responded the best.
Yeah, and I think this was a good example of different people responding to different things, because I've had a lot of patients. We ran the trial, and we had a lot of patients on the CBD through the open label extension after the controlled trial, and then we've had other patients that got the medication online, and some patients really do have a nice response to it, I think, and a persistent response, and then others really don't have much response or actually kind of get worse on it. And so it's like a lot of different behavioral medications in Fragile X. There's a variable response, but if even 20 or 30% of patients have a nice response to it, you'd like to have that in your armamentarium to be able to treat patients, because that's a very significant thing. to be able to add to what we have currently.
Can also help the mothers too. They can sleep better with CBD. I've had many mothers tell me that their irritable bowel syndrome and many other symptoms and anxiety got better with CBD.
Yeah, now there's some older drugs that are being retested, right? Our baclofen that was a subject of one of the first studies that we were involved with. I know that that's now been repurposed or it's coming along again. Is that where?
Yeah, our baclofen was acquired from the Simons Foundation, which acquired it from Seaside. And the new company that has it does plan to move forward and try to work through the FDA with it. That was another really good example of a drug that works really well for some people, but does not work for everyone, but there's, 40 or 50% of patients that really had a pretty nice response to it, as well as you can tell. I mean, we ran several large controlled trials, and actually the pediatric trial did show benefit on the measure of irritability and on a measure of parenting stress that corresponded to each other. They kind of narrowly missed the primary outcome, which was social anxiety. But again, you don't always quite know what to make the primary outcome when you're targeting the whole Fragile X. So that trial has never actually been presented to the FDA because Seaside went out of business before it was completed. So the new company, the old database exists, and I believe there's a plan to run through the data that exists and determine what needs to be done to try to bring our baclofen to an approval in addition to what's been done before. So yes, it is reemerging, but we don't know yet what we will need to do to cross the bar and make it available to patients.
Oh yeah, I was going to say, I mean, we think, and through some of our single dose and some of our chronic dosing with GABA modulators generally, that we see different responses based on protein status. So, for example, females that are making below average, but readily discernible, very low protein males, zero protein males. So we think that, molecular status will aid us in parsing our interpretation of GABA modulation and potentially other small molecules in Fragile X to sort of personalize or link, the subgroups that are best responding. And I think that's been a common theme of this discussion, that, we've had potentially success in the past that was married to, within the same trial with those who were not responding. And I think now we're really beyond that through our ability to characterize the patient populations better.
Yeah, I've had several families that told me that our baclofen was the best drug that their child has ever been on.
Oh yeah. We have families who are still pining for that drug, and they come into the clinic and they're like, what's happening? Are we ever going to be able to get this drug again? And they remember that. They've been on many things since that time, and they still remember that as the best time period for their child with Fragile X. So I think there are some who really had a substantial response, and it would be great if we could make it available for those patients.
So there are several ongoing trials that, you're aware of, I'm aware of. Maybe, Liz, you could summarize where you see some of these going and when they'll be reported out.
Well, right now, so the CBD process is ongoing with a new trial, because since the response was only in the fully methylated patients, the FDA wants a replication of that. And so the company is doing another trial. And then there are a couple that are in setup. We have an ongoing trial that is funded by NIH through the NeuroNext network, and Rondi and Craig are both sites on this trial. And this trial was an attempt to look at the impact of a drug that was meant to be improving synaptic connectivity, and also push the patients to learn through an intensive learning intervention to measure whether we could amplify learning with the drug in a specific area. So we're studying language learning, and we're using one of the mGluR5 blockers that failed for behavior in adults and adolescents with Fragile X. But we're enrolling very young patients who are, who would be learning at the most rapid rate because they're young. So 3 to 6 year olds. And then because 3 to 6 year olds with Fragile X are really learning language at that age, we're doing an intensive language learning intervention. And at the patients are randomized to drug or placebo, and we're looking at, will the drug amplify the language learning if we really push kids to learn, rather than just having them have background treatment. And so that trial is not quite over yet. It's going to end at the end of August, and then we have to lock the database, and we'll get some idea of whether this strategy was effective or not.
Very good. And Rondi, I know you've been very enthusiastic about metformin, the generic drug that's been around for a long time.
Yeah, so metformin does lower the mTOR pathway that's too elevated in Fragile X syndrome. So it's a targeted treatment too, and inexpensive. And we've done a controlled trial at 3 sites, not just at the Mind Institute, but 2 sites in Canada. And we will finish enrollment in sometime in August. So by the end of the year, it's a 4-month trial. By the end of the year, we can look at data from all 3 centers. We've seen very good anecdotal data for individuals that were treated with metformin clinically. We've seen some improvements in IQ in a couple of individuals, and the inhibition of macroorchidism or large testicles when it started before puberty, but we don't know if that occurs in all individuals. But we will also, after the control trial ends from a 4-month treatment, we'll have longitudinal data for those that are on a year or 2 years. So we'll see over time if it does improve IQ.
Great. I think one of our, one of the purposes of this is to highlight Fragile X Awareness Day on the 22nd. And I know that FRAXA has been a major driver of pushing awareness of this, as well as sponsoring many of these preliminary drug trials. So maybe you could just give a little, shout out to FRAXA, or tell us what you think of their role in the Fragile X field.
Yeah, so I think for a long time, FRAXA has spurred on research that was needed and wasn't necessarily at a point where it could get funded by NIH, and has brought a huge number of neuroscientists into the field back in the 1990s when it was more about genetics. And from that has emerged all of these mechanisms that we have in the mouse. And then they've been very supportive of getting animal studies done with new compounds, as well as funding early trials, like the trial that we talked about at the beginning of this with the phosphodiesterase inhibitor. And so they've been really key to moving research forward in this field at a speed that I don't think would ever have happened with just NIH funding, for instance.
I mean, FRAXA really got me into this field. My first grant was from FRAXA to study medication in Fragile X. They've been serially supporting, high risk, high reward work with, using available drugs, repurposing them in Fragile X. They've been invaluable to our gene therapy efforts. And really, it's a go-to location for, as Liz said, nimble and astute funding opportunities at an early seed level to launch larger programs. I mean, I don't think we would be doing our NIH center grant work now if not for FRAXA's support of getting us going in the past. And I think we all the 3 of us and many thought leaders have constant dialogue with FRAXA. I mean, it's really a back and forth collaborative process, and it's incredibly supportive. And I don't think we would have our success as a center and our success in the field for the stakeholders without their input and support.
Craig, can you say a little bit about the committee of the National Fragile X Foundation's Committee on Drug?
So several years ago now, the National Fragile X Foundation put together a clinical trials committee that I've been lucky enough to chair, and Liz and Rondi and many others are members. And we really exist to support treatment development broadly in the field. So we're available to advise, provide consultation to industry, to academics, to anyone, to try to improve and inform from the earliest stages of drug and treatment development to improve things like signal detection, thinking about outcome measures, population enrollment. A lot of things we've talked about today in detail are available from that committee and our partnership with the National Fragile X Foundation to bring various stakeholders together with thought leaders, because we all want to de-risk and see success. So I think the foundation was really forward thinking in coming up with a mechanism to bring thought leaders and stakeholders together so we can have an advisory role, hopefully sooner rather than later. And that's what we've learned, and that's what the committee is available for, to support all comers.
I think the
Great.
National Fragile X Foundation has done a wonderful job in every other year international conferences and establishing Fragile X clinics throughout the country, but also internationally. So it's the international conferences, which have typically taken place in the US and maybe in the future may take place in other countries, but it's been marvelous to bring families and parents and therapists of all different types, special education teachers, speech and language, OT therapists, together with basic scientists, so that families and scientists can intermingle, share ideas, and talk about advances. So this is a great conference if you haven't been to one yet.
Great.